Now moving to another important component of the periodontal ligament. Before this we have studied about the periodontal fibers and which type of collagen is present in those fibers. Now moving towards the cellular element of the periodontal ligament. So let's start it. Bismillahirrahmanirrahim. So first of all, we have four types of cells in the periodontal ligament. First, connective tissue cells, epithelial rest cells, immune system cells, cell associated with neurovascular cells. These cells now we will study one by one. In connective tissue cells, we know in connective tissue we have fibroblasts, cementoblasts, and osteoblasts. Fibroblasts form collagen, then cementoblasts form cementum, and then osteoblast forms bone. Now fibroblasts are the most common cells in the periodontal ligament. If in your viva they have asked you which cells are present in mostly in connective tissue of the pedials, they are fibroblasts. They are paired to ovate or elongated. Either they will be ovate or elongated cells oriented along like a principal fibers. They show a pseudo like processes, pseudopodia like processes. Okay, these cells synthesize collagen and possess the capacity to phagocyte, means they form the collagen. Second, they have the property of phagocytosis, hold collagen fibers and degrade them by enzymes hydrolysis, means they actually degrade the old collagen fibers and through their enzymatic hydrolysis processes. Okay, so in connective tissue we have most fibroblasts. The fibroblast synthesize the collagen and phagocytose the old cells. The collagen turnover appears to be regulated by fibroblast. Okay, they are saying that the collagen is actually their turnover is regulated by fibroblasts in the process of intracellular degradation of collagen that do not involve the active collagenase. They are saying that fibroblasts basically actually regulate their process of degradation of collagen, in which the collagenase action is not required, in which this enzyme is not present. So the fibroblasts then regulate by themselves their degradation. Phenotypically distinct and functionally different subpopulations of fibroblasts exist in the adult periodontal ligament, means they are saying that different type of fibroblasts are present in the periodontal ligament and they are appeared to be identical at both the means you can see through the light and electron microscopic levels, but they have different function. Means in periodontal ligament we have different type of fibroblast and they have actually different functions, like they have secretion of different type of collagen types and production of collagenase. Osteoblast, cementoblast, and cementoblast and osteoclast and odontoclast are also being seen in the cemental and osseous surfaces of the pedial, but the most of them was fibroblast. They synthesize the collagen and also degrade them. Actually basically the epithelial rest of malaise forms an latest work in the periodontal and appear as the either isolated clusters of cells. So basically they are appear as a isolated cluster of cells or interlacing strains depending on the plane in which the microscopic section is here. Continuity with the junction epithelium has been suggested in the experimental animals. These epithelial cells are the remnants of Hartwig root sheet, which actually disintegrates during the root development. Okay, means the rest cells of malaise are present in the apex area of the pedial. Okay, through which also we have the development of radicular cyst we have studied. Epithelial now moving to epithelial rests are actually distributed close to the cementum throughout the pedial, means they are saying that the epithelial rests are basically distributed close to the cementum throughout the pedial. They are mostly numerous in the apex area and cervical area. They diminish in number with the age, mean they are actually degenerated with the age. Their degeneration and disappearing or by undergoing calcification to become cementicles. Either they degenerate or converted into the classified mass is called cementicles. These epithelial rests. Okay, these cells are surrounded by the distal lamina, basal lamina. They are interconnected by hemodesmosomes and they contain tonofilaments. They have also hemodesmosomes means they are interconnected by hemodesmosomes and filaments. The functional properties are still considered to be unclear. These epithelial rests are reported to contain keratin growth factors, and they have also shown have positive for tyrosine kinase A neutrophin receptor. Okay, so they think that these epithelial rest cells also has keratinocyte growth factors and tyrosine kinase A neurotrophin receptor positive. In addition, these cells can proliferate when stimulated, and they participate in the formation of periapical cyst or lateral root cyst, which I have told you actually. Defense cells or immune cells are also present in this PTL that include neutrophils. They have lymphocytes, macrophages, mast cells, and eosinophils. These cells are also associated with the neurovascular element which is are similar to those cells that are found in other connective tissue. Okay.